Hello everyone, this is the Science 30 version of DNA replication and protein synthesis. And I say the Science 30 version because this is really toned down compared to what you would get in, say, Bio 30. DNA replication and protein synthesis, they are in fact very complicated processes with many steps and many different enzymes that are involved in these processes. But as far as Science 30 goes, fortunately, you don't need to know really too many of the details at all. So as we go through this presentation, I will be touching on some of the steps and some of the details. But again, at the end, as far as Science 30 goes, it's pretty straightforward in terms of what you do need to know for DNA replication and for protein synthesis. So let's start off by going back to the DNA, the structure of DNA, because that is where we are going to for both the DNA replication and for the protein synthesis. So the left-hand side picture, the structure that we see here of the DNA where it is twisted, this of course is referred to as the double helix or double helical form. If we do take that and just unwind it, as it's showing in the middle here in B, it's a little bit easier and straightforward to talk about the different components. So how do we go from this straight ladder to the DNA double helix? Well, if you just hold on to one end and twist the other end, then that does generate this double helix. So DNA is a polymer, which means it is made up of repeating units. It is also called a polynucleotide, because the repeating units, they are nucleotides. And I'll write that at the top here as well. Nucleotides are the repeating units, and nucleotides are made up of three things. They are made up of a sugar, the deoxyribose sugar. They are made up of a phosphate, and they are made up of what's called a nitrogenous base. So these ones here, the sugars and the phosphates, they make up the sides of the ladder or the backbone, whereas the bases, that's where we do find in the middle here. So in terms of the sides, they are really just providing a structural role. So we have alternating sugar, phosphate. And what is coding for the genetic information are the A's, the T's, the G's, and the C's, which are the nitrogen bases. So keep in mind as well that here what we have are 8 steps in the ladder, but DNA, in fact, the shortest of all of the human chromosomes, they are millions of these base pairs in length. So this goes on and on and on in this direction, and on and on and on in the opposite direction as well. So in terms of the nucleotides, let me just circle a couple of them here. So each one of these blue rectangles will be a sugar and a phosphate. And then, of course, we have the letter, which is the nitrogen base. So this is one nucleotide. This would be the next nucleotide. And they are held together to the complementary base of the nucleotide on the other strand of the DNA. So those are held together by what are called nitrogen bonds. So we can see that these two backbones, they are running parallel to each other, but in fact they do run in opposite directions. So if one of these strands, the one on the left we'll say, is read going from top to bottom, or written going from top to bottom, the other one is parallel, but it is read and written going in the opposite direction. At the right-hand side, what we can really tell is, or see, is that it's really just all chemistry that we're talking about. Every single one of these nucleotides, the sugars, the phosphates, the bases, they are just made of elements from the periodic table. So the nitrogen, the hydrogen, the oxygen, the carbon that are from the periodic table. And we can see that running down the middle here, these dashed lines, those are the hydrogen bonds that are holding the two sides together. And also with these arrows, it again points out that even though the strands are parallel to each other, they are running in opposite directions, or what we call anti-parallel. So when James Watson and Francis Crick came up with the basic structure of DNA going back to 1953, when they did discover it, it was already kind of clear that there was a possible mechanism for duplicating the DNA. If we were to take and break all of these hydrogen bonds running down the middle, which is pretty easy to do, and then separate the left-hand strand from the right-hand strand, there's only one other thing that can attach to this thymine, and that's another adenine. there's only one thing that can attach to this guanine, and that's a cytosine. So because of the complementary base pairing, it was already clear that if we do separate the two halves of the DNA ladder, then what we have are templates. We have a template on the left, and we would have a template on the right as well. And those templates would serve as attachment points for the complementary bases. So that is necessary for this process of DNA duplication, or what we actually call DNA replication. That is DNA replication. So why is DNA replication so essential? Why do you need to make copies of the DNA, copies of the chromosomes? Well, every single time your cell divides, you need to copy the DNA. So each new cell gets exactly the same genetic information, or at least that is the case if we are talking about mitosis. So every single one of us, we started like this. We were one single cell, and in that one single cell, we did have 46 chromosomes. They're arranged in pairs. 23 came from your mother, 23 came from your father. And then what happened is that one single cell divided into two. And each of those had not half the number, but exactly the same number of chromosomes. And then these ones divided into two, and this one divided into two. And all along the way, all of these cells of yours have 46 chromosomes. So what that means is that before we have cell division, before we divide up the chromosomes, what we need to do in between here is we need to duplicate, or we need to replicate the chromosomes. So if you think about in your body right now, it's not just when you were an embryo and a fetus and you were going through all of these stages of embryonic and fetal development, but even in your body right now, you have cells that are dying and need to be replaced all the time. And you have a reservoir where you can replace these cells, what are referred to as stem cells. And in your skin, for example, your skin cells are dying all the time. You need to continually replace them all the time, which means continually you are always replicating your DNA. You're making exact copies of it, or as exact as you possibly can, before your cells actually go ahead and divide. Now remember, there's another kind of cell division as well, and that is meiosis. So whereas mitosis is for your body cells, it includes any of the cells in your body of the 250 or so different kinds of cells, but meiosis is only for one thing, and that's for producing the sex cells, the gametes. If you are a male, of course, those are the sperm that are produced in the testes, and if you are a female, those are the eggs that are produced in the ovaries. So it's not the process of mitosis that is used here. This is the process of meiosis. And now each one of those sperm and eggs, instead of ending up with 46 chromosomes like all of the other cells in your body, they only end up with half that number of chromosomes. So now when we do have a sperm cell and an egg cell that come together, each of them have 23 chromosomes. They unite to form the first cell of a new organism. So again, this was you back when you were one single cell, and this is what is referred to as the fertilized egg or the zygote. So again, the point here is that before you can go ahead and generate the sperm and the eggs, you need to first of all replicate the DNA. And that has to be done with very high fidelity, with very high accuracy, very, very, very few mistakes. So it needs to go at an incredibly rapid rate. So keep in mind that of each of these here in the sperm, it's 23 chromosomes, but it is made up of 3.2 billion steps in the ladder. In other words, 3.2 billion base pairs, and that needs to be replicated. So how does this work? So overall, it's pretty straightforward. So each strand of the DNA, it's going to unwind. We don't want a tangled mess. And then we're going to use each half of it when they separate as a template for the formation of the new complementary strand. So a reminder just again about the complementary base pairs. Wherever you have a T, what is complementary is going to be an A. Wherever you have a G, what is complementary is going to be, oops, not a G, but it's going to be a C. So if we have a strand that is C, T, T, A, C, G, and if this is our template, the only bases that can attach to this are the complementary bases. So here we would end up with the G, A, T, G, C for the complementary bases. So this is important for you to understand in terms of DNA replication and the complementary base pairing. As soon as you have those two halves of the DNA double helix, the templates that are folded apart. I did mention that it's a fairly complicated process, so it does require energy. It of course requires pre-existing DNA to act as a template. It requires a huge reservoir, and we are talking billions of nucleotides for replication to take place, and it takes several different proteins and enzymes, of which for Science 30 you don't really need to know anything about. So steps in DNA replication. So let's break it down a little bit in terms of what is going on, and then I'll show you an animation here as well. So first of all, we have the unwinding and the unzipping of the DNA double helix, and that's going to expose the templates, the single-stranded templates that have the bases that are now not attached to anything else. But they don't like that. They want to be attached to some other complementary base, the complementary nucleotide. So enzymes are going to bring in the free-floating bases. It's really the nucleotides, of course, with the bases attached to them, and they're going to attach to the complementary base pairs on that single-stranded template that we created when we separated the DNA. And again, we see the complementary base pairing indicated here as well. The sugars and the phosphates that form the backbone, they're going to join together. They're going to form a covalent bond. So we have a nice continuous polynucleotide. They're held together. They're bonded together. So we started with one single DNA molecule, and now we have two identical molecules of DNA that are formed. So let's take a look at an animation here. I'll just stop this for a second because it's not pulling up. Okay, so here we go with the animation. So here we're taking a look at a cell, and it's a square in nature, so it would be a plant cell in this picture, but it doesn't matter if it's a plant cell or an animal cell, the process is essentially the same. The green circle, it has identified as the nucleus, and the chromosomes, the black bars that we see in the nucleus, they've simplified this, so instead of 46 that we would find in a human, there are only 6 here. So we have the pairs, we have 2 short ones, 2 medium length ones, and 2 longer ones. So this would be a diploid cell because we do have 2 copies of each one of the chromosomes. So they're just going to, in this animation here, zoom in on just one small section of one of the chromosomes so we can see what is going on with replication. So here we have about 10 base pairs, and we can see the checkers along the side, the black and the white, representing the sugars and the phosphates. And the colors that we see, the green, the yellow, the red, the blue, those would be the nitrogen bases. So in the case of this model that we're taking a look at here, this picture, we do have the DNA double helix that has already been unwound. So this is just the straight ladder that we're taking a look at. So what happens with DNA replication, and really again for Science 30, it's not much more than this, is you're going to separate the 2 halves. Now what we have are the templates from the original strand, and these exposed bases, they don't like to be unattached. They want to attach to something else. So this is where we need this huge reservoir of a whole bunch of the nucleotides that are available, and then enzymes are simply going to bring in and attach the complementary bases, the only one that can attach to them, the complementary bases. So we can see down at the bottom here, we do have one of these, they call it the subunit, but this is going to be one of the nucleotides. So in yellow here, that's the nitrogen base, and we can see that that is going to be complementary to the green one. So perhaps the green is a T, and the yellow would then be an A. So we're going to bring that in, and we're going to do not only the same thing with the left-hand template, but we're going to do it with the right-hand template as well. So now if we take a look at these are our two new DNA molecules. They are comprised of one half of the original strand, and the other half entirely new from these nucleotides that were brought in. But of these two DNA molecules that we have, if you take a look at all the colors, what you can see is that they are identical. And that is the point of the DNA replication. So now we're just going to zoom out. We have replicated that small section of one of the chromosomes, and now that's going to take place with the rest of that chromosome and with all of the other ones as well. So now what it's representing in this picture is we have these bars that are doubled up, which are the duplicated chromosomes that we now have after the DNA replication process. And this picture here nicely summarizes, once again, truly everything that you need to know science theory-wise. This, again, is our DNA double helix. Here we have only one single strand. If we jump over way to the right, here we have one here, and we have another one here. So we have two. So we have duplicated or replicated the DNA. So it shows that right at this point in the middle, that's where we're unwinding the DNA. We can see all of these free nucleotides that are coming in, and they're attaching to the exposed bases, the exposed nucleotides on both of the template strands. So as this fork here opens up continuing to the left, we just bring in more and more of the nucleotides, and as they're brought in, they form the complementary bases that we can see all of these have formed. In the end, we just need to retwist the DNA. We started with one, and now we have two. That is DNA replication. So the flow of genetic information and protein synthesis. So almost every imaginable process that you can think of that takes place in your body does involve protein. So I'll give you a whole bunch of examples here. But now the discussion is going to be about how that genetic information is used. In other words, the DNA, to ultimately make proteins, and the making of something is called synthesis. So we're going to use the DNA in order to make the proteins. DNA provides the code, and the proteins are what we consider to be the so-called workhorses of the cell, because they are carrying out, almost every imaginable process that you can think of that takes place inside of your body. So a number of different examples here. So we've seen this one already. Inside of your red blood cells, you have a protein, hemoglobin, that's function is to transport, well, primarily oxygen, but it's also carbon dioxide as well. So it's not the DNA that does that, it is a protein that does that, and that protein is hemoglobin. The instructions for making the hemoglobin are housed in the DNA. Number of proteins, they are structural in nature, so histones are what DNA wraps around to condense it and keep it in a fairly compact form inside of your cells and inside of the nucleus. So various different structural proteins. Enzymes, thousands upon thousands of enzymes in your body. They catalyze reactions. They speed up reactions inside of your body. And all of your digestive enzymes, what is breaking down your food, the fats, the carbohydrates, the proteins, it is going to be an enzyme, which is a protein. And one example here is pepsin, which is in your stomach, and it actually breaks down proteins that you take in through your diet. When your muscles contract, there are multiple different proteins that are involved in the contraction of your muscles, whether it is your heart muscle or your skeletal muscles that are attached to your bones. Defensive antibodies that we've chatted about as well, they are proteins. They attach to antigens and, of course, aid in the destruction of anything foreign that gets into your body. Signaling molecules, how cells communicate with each other, various different mechanisms, but hormones, they are one of those mechanisms. Insulin is responsible for regulating your blood glucose, and that is a protein as well. And energy. Proteins can be broken down to provide your cells with energy to make ATP to carry out different cellular processes. So proteins are not synthesized directly from DNA. So this is where I'm going to go a little bit beyond Science 30, because Science 30 actually does want you to think that proteins are made directly from DNA. But I'm going to give you a little bit extra. I'm going to complete the picture for you, because it's just simply not true that proteins are made from DNA. So I'm going to give you a little bit more information, and I'm going to talk about something that's abbreviated RNA, which stands for ribonucleic acid, whereas DNA is deoxyribonucleic acid. So truly, in order to make proteins, you do need RNA and what is abbreviated M, or messenger RNA. So remember that DNA, where it is found, is in the nucleus. This paler yellow color that we see here, that is in the nucleus. So yes, you should realize that that is where DNA replication is taking place, is inside of the nucleus. In the past, you have heard about ribosomes, various different organelles inside of a cell. Ribosomes are the protein factories, but where they are is in the cytoplasm. They're not in the nucleus. So we have a little bit of a problem here. DNA, which has the instructions for how to make a protein, it's in the nucleus, but it can't leave the nucleus. Proteins have to be made in the cytoplasm. That's where the ribosomes are. But how do they get the instructions if the instructions are stuck in the nucleus? Well, this is where this RNA comes in. So whereas the DNA is big, it's long, I'll just put here DS, it's double-stranded. RNA is shorter, and it's only made up of 1/2, or single-stranded. So because it is smaller, it can actually go out of the nucleus and into the cytoplasm. So let's just say that the protein that we want to make is that insulin that's responsible for regulating your blood glucose. It can't be made directly from the DNA. But somewhere amongst those 23 pairs of chromosomes are the instructions for making insulin. So what you need to do is make a copy of just the information for making insulin. So we'll just call this insulin info, only for how to make the insulin. Whereas in your DNA, it's the instructions for all of the tens of thousands of proteins that your body needs to make. So you just need to make the insulin, so you make a copy just of that information. It's shorter, it's single-stranded, it can go out of the nucleus into the cytoplasm, and now that's where the ribosomes are, and that's where the protein can now be made. So that is truly how this process does play out. Don't worry about these terms, but they are transcription and translation. They're just the different components of protein synthesis, but you don't need to know those names for Science 30. However, this is kind of what it looks like. So this is the making of the messenger RNA. It takes place in the nucleus. It's kind of like replication, only different enzymes are involved. You get RNA instead of DNA. It's single-stranded instead of double-stranded. And there's one base that's a little bit different. Okay, again, don't worry too much about those details. So here we have our double-stranded DNA. So right in here, we have 1/2 of the strand of DNA, and here we have the other one. So here, this might be the instructions for making insulin, but you cannot use that to directly make our protein. This is our protein over here. Proteins are made up of amino acids, and that's what these names are. Those are the names for individual amino acids. So what you need to make first is this that's right in here, and it is abbreviated mRNA, which stands for messenger RNA. So it's this that's going to take the instructions for making insulin out of the nucleus and move it into the cytoplasm, where the ribosome factory is, which is the ribosomes. So now, what do you do with that messenger RNA? Well, we're going to get the reading of the DNA. Sorry, this is going back to transcription. The reading of the DNA. We're going to make the messenger RNA, and then we're just going to take that information into the cytoplasm. So let's just see what it looks like here, just to give you a complete picture of what is truly going on. This is our double-stranded DNA. It has been unwound. It's really quite straightforward and simple, at least in terms of overall the process, and that's what this animation shows. So we're going to unwind the DNA, pry it open. We're going to read one strand, and we're going to use that to make our single-stranded messenger RNA, and that's what we see coming out of the bottom. So that in itself isn't overly complicated. So now we can see that the DNA is bigger, it's bulkier, it's longer, it can't leave the nucleus, whereas this is small enough that it can leave the nucleus. So now that that's in the nucleus, now we're just going to use it to make the proteins, and that's a process that's called translation. So in this picture here, this is our messenger RNA that's now in the cytoplasm. And now we just want to link together a whole bunch of these amino acids. And this molecule is going to do the decoding. It's going to read the messenger RNA and insert the appropriate amino acid. So what is being made at the bottom here is called in this picture a polypeptide, but a polypeptide is a protein. Okay, so the point here is that in order to make a protein, really this is what you need to go. You need to go from DNA to messenger RNA and then to proteins. In fact, this is called the central dogma of molecular biology. But again, Science 30 doesn't want you to know the central dogma, I guess, of molecular biology. They just want you to go from DNA to the protein. So after all of those, and again, it's not really all the details that I did go into, but after giving you a little bit more information in terms of what truly does happen, let's take a look at what you actually do need to know for Science 30. I'll just show you a couple of quick little animations here. So this one is translation. So this is using the messenger RNA to make the protein, and it's going to be similar to that previous one that I showed you. So this is our single-stranded messenger RNA now. This is our decoder, and this is an amino acid. And we're just going to read the messenger RNA. It's actually read 3 bases at a time. And we're linking together amino acids. So what's being made at the top here? Where we're joining together. Here we have 3 of these amino acids. Now we have a 4th one coming in. So each of these ones, where it has the MET, the LYS, the PHE, and the GLY, those are individual amino acids that are forming the protein. And just one more here to give you the overall picture, a better picture of what is truly taking place. We're going back to that picture that we saw before with the replication, only now this is for protein synthesis. Now, we don't need to use all of the DNA. We only need one specific part of it. So what they're zooming on here, again, this is maybe the instructions for making hemoglobin or insulin or that protein pepsin. What do? You separate the DNA, kind of like replication. Only now we're just going to read one side. And what's being made here is not DNA, but that's the RNA that's being made, and that's the messenger RNA. DNA just snaps back together. It can be used over and over again, but we just made this RNA. So what happens to that RNA? It's small, and it can leave the nucleus. Zip out through the nucleus into the cytoplasm, and now we're going to make a protein. This is our messenger RNA. We're going to read 3 of these bases at a time, and we're going to join together amino acids, joining them with what's called a peptide bond. So this is our protein that we've made here, a whole bunch of amino acids that are linked together. In the real world of molecular biology, there is what is called an RNA codon table, a messenger RNA codon table, but not in Science 30. In Science 30, we have a DNA base triplet table. So this one here, it shows us 64 different possible 3-letter combinations, and each of those 3-letter combinations in DNA codes for a specific amino acid. So what this says is if you get these 3 letters together in DNA, TTT, that's for the amino acid phenylalanine. If you get TCA, that's for the amino acid serine. If you get AGA, that's for the amino acid arginine. You might also notice here that even though there are 64 different possible combinations, there are actually only 20 different amino acids. So for some of the amino acids, there's more than one code. So TTT and TTC, both code for phenylalanine. All 6 of these all code for leucine. All 3 of these all code for isoleucine. There are also 3 of them on here that don't code for any amino acid, but what they code for is that this is the end of the protein. They are the stops. So the TAA, TAG, and TGA, those are all what we call stop triplets in the DNA. And one of them should always be at the start. ATG is the start triplet, and it does code for amino acid, and that amino acid is called methionine. So let's just go through and read this table and get a little bit of practice. So what we're given here is a sequence of DNA. And what I like to do is just divide it up into the 3 different bases, because it's 3 at a time that you're going to be using for reading that triplet table. So this is our amino acid sequence, and what we want is the amino acid sequence. So we just need to go back to our codon table and find ATG. So if we go back here, ATG, in fact, is methionine. So what would you do? Well, we're just going to write down here that the first amino acid, I'm just going to use the abbreviation is methionine. And then I'm just going to form a bond here because this peptide bond, this line, is going to join it to the next one. So CCA. What is that one? So it does have it organized here. The first base is along the left-hand side, the second base is along the top, and the third base is along the right-hand side. So you can go by that. Sometimes it's easier just to scan across the table to find it. So CCA is what we're looking for. So CCA, this one right here. So that is proline that we're going to insert in for our second amino acid. Go on to the third one, AGA. So AGA is going to be this one right here, the arginine. And we'll just do one more, the TTG. TTG is going to be our leucine that we would have here. Okay, so you're going to join those together. Those would be the amino acid sequence, but this is also the protein that you are making. You may also need to go in the opposite direction. So with this one here, we're given a number of different amino acids, and you're asking what could be the possible DNA sequence. And it is the possible because, again, there might be more than one triplet that codes for alanine. So usually what I say is just pick the first one that you do come across and write that one down. So let's find alanine on our table here. Okay, so we'll just sort of go down the rows here. Oh, here we are, alanine. So I'm just going to pick that first one, GCT. GCT. So those are our first 3 letters, our first triplet. Then we have serine for our next one. Again, kind of going down. Even though there are a bunch of them, again, I'm just going to pick the first one here. So TCT. TCT, and again I'll just do one more, and that is isoleucine. We'll go back up here one more time, find our isoleucine, first one right here, ATT. We'll go with that one. So it is a 3-to-1 ratio. We have 3 DNA nucleotides to every 1 amino acid. So what that means, if we have 1, 2, 3, 4, 5, 6, we're going to times that by 3, and in the end you should be giving me 18 letters in this sequence here. For the stop, yes, there actually are 3 more for that one, so we should probably add in 3 more. There is not an amino acid, but there is a stop triplet. So in fact, it would be 21 letters that you would be giving me here. And if we go up to this one, so we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I don't know what this one is, but let's just say that it is a stop, and that would mean that there's no amino acid for the stop. So if we have 10 of them, and we have 10 times 3, so that again is 30 of the bases, but it's only going to be 10 amino acids that we would have here.